All right, everybody, welcome to Friday Compass Seminars. Today we have Kaycie Lanpher, who is giving a presentation, her 1-hour pre-defense seminar. Kaycie graduated with a BS from the University of Texas at Austin. She is my office mate, and she won Best Student Presentation last year, so this should be good today, although no pressure. It's a much longer talk this time. And today she's going to be talking about marine microbial metabolic responses to environmental conditions, the impacts of light, temperature, and oxygen concentration. All right, Kaycie, take it away.
Thank you, Anne. I appreciate that. I will start sharing my screen now. Okay. Everyone should be able to see. Please let me know if you don't see my presentation. So as Anne mentioned, I will be talking about marine microbial metabolic responses to environmental conditions, the impacts of light, temperature, and oxygen concentration. Okay. So today's outline, we're going to go through some background information about marine microbes and the Adenylate energy system. I'm going to go through the questions and hypotheses we're going to cover, the methodology, and then get into the environmental studies with the temperature, light, and oxygen. The marine microbial organisms, or microbes, are organisms in the micrometer size range, so they are quite small, and they are very diverse. So this includes phytoplankton, bacteria, archaea, protists, viruses, and the diversity of these also means that there's a strong diversity in metabolism as well. So we're looking at this whole community when I'm saying microbes. The role of microbes in the ocean in global elemental cycles, they have three main roles that I'm thinking about. The transfer of large amounts of material from one compartment of Earth systems to another, the transformation of this chemical material in form, so altering its reactivity, its fate in the ocean, and then they're also able to respond rapidly to perturbations of the system, so changing oxygen, temperature, light. Those also change their metabolic controls on these elemental cycles. This image on our right is an image of the seawater from Kim. They're DAPI stains, which means I've stained their DNA. So this is actual images of microbes, and they're quite abundant in the marine environment, so 10 to the 8th cells per liter on average. So they are, even though we can't see them with our naked eye, or most of them, they are very abundant and have strong controls on global cycles. So what that looks like in the ocean is they are interacting with a lot of different nutrients and different roles, but I'm going to focus on the fact that they transfer gases, specifically oxygen and carbon dioxide, through the system. And with carbon dioxide, that's taken up by these microbes, especially phytoplankton, and then further transformed into particulate organic matter or dissolved organic matter. So this specifically impacts the role of the ocean as a carbon sink, or the export of carbon into the deep ocean by these microbial organisms. And the microbial activity, the individual activities of all those diverse organisms, really is what's controlling this amount of sink, or the flow of oxygen and other nutrients in the environment. So we want to understand the microbial metabolisms to further understand the role that they have and their impacts on these metabolically controlled cycles, the biogeochemical cycles. If we're looking at microbes as a whole, all microbial organisms require three main nutrients: carbon, nitrogen, and phosphorus, at about a ratio of 106 to 16 to 1, or the Redfield ratio. So I'm going to focus on phosphorus for this talk. So phosphorus, or P, is depleted in a lot of our large ocean basins and is potentially limiting for biological activity. So the phosphorus in this image on the right, the very like white or light purple shows where it is low, especially here in the basin for the North Atlantic, which is part of my talk that I'm going to go over is experiments that happened here. And it's also low in the South Pacific gyre, and part of my talk goes into data from here. So whenever I'm talking about P limited or the phosphorus dynamics, we're like looking in oligotrophic areas. And because we're in these oligotrophic environments, phosphorus can be an important lever, or that's controlling the change of these microbial productions or activity, in turn controlling those rates and transformations of carbon, nitrogen, phosphorus, oxygen. And so understanding the interactions between microbes and phosphorus can be very important to understanding their overall roles in these other cycles. When I'm talking about phosphorus stocks, I'm going to be focusing on three main large pools. So we have on the left your dissolved phosphorus or dissolved inorganic phosphorus in the form of orthophosphate or phosphate. And that's most commonly what we think of when we're talking about phosphorus in the ocean, and nutrients. Most of the time we're talking about phosphate or inorganic phosphorus. We also have dissolved organic phosphorus, which is more complex in structure and includes phosphonates and phosphoesters. And those are the dissolved organic molecules that are present in microbes in particulate phosphorus, so in the lipids, DNA, RNA, metabolites, ATP. So if we're looking in the oligotrophic ocean, this is what our general pool size is. So the sizes of those different phosphorus pools in the ocean. So we have our inorganic phosphorus, that DIP, organic phosphorus, and as you can see, the organic phosphorus is much larger in size of the pool, which means there's more organic phosphorus available in those oligotrophic oceans than the inorganic. And this inorganic is what I was showing on that plot that described where it's oligotrophic and not with phosphorus. And in our particulate phosphorus, we're looking at the particles, so the actual microbes, these single cell organisms that include phytoplankton and bacteria and archaea and viruses. And so looking at the actual particles is the phosphorus stocks. So whenever I'm thinking about activity within these phosphorus dynamics, I'm looking specifically at the adenylate energy system, which is important for tracking energy in microbial metabolisms, and it includes adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, and that inorganic phosphate. So here is, if you've been to one of my talks before, this might be familiar, this ATP or adenosine triphosphate. So we have the adenine, ribose, and then the 3 phosphate groups. And inside microbes, they hydrolyze this final phosphate group to release energy for metabolism, thus creating ADP and this inorganic phosphate. And so using this system, we can track the storage and then release of energy in that microbial organisms. And some of the really important and valuable things about this adenylate system is it's ubiquitous. So that means all microorganisms, all organisms for that matter, they use this adenylate system for energy. And so it is very, very important, and it can be used to understand all microbial organisms. Also, almost all metabolic pathways within the microbes rely on it. So not only do microbes all have this system, but they all rely on it for these metabolic pathways. What that tells us is that this can be used as a broad scope, or like painting with a broad brush across microbes, and it's powerful to enable a quantification of important fluxes by the microbial community as a whole across many environments. So whenever we're bringing it back to that phosphorus, what we can do is then the phosphorus, which all microbes need, again, is taken up. Instead of just looking at the phosphorus going into microbes and having that pool, we can see what they're doing with it and how that impacts their metabolism. So looking at the phosphorus going into ATP, which then impacts metabolic processes, because that's where they get their energy for those processes, which then causes changes in their fluxes and doing the export of carbon or producing oxygen. So this really helps us to determine what the microbes are actually doing when they're impacted by phosphorus concentrations. So our questions that we're really interested in is broadly just how do microbes respond to changing environments? When I'm thinking of how do they respond, like how are they changing their phosphorus uptake or their phosphorus interactions and what that actually means on their metabolism. So for our three main different environments that we're thinking about, or different test like conditions, I hypothesize that microbial organisms will increase their metabolic rates independent of growth rates under elevated temperature. I also believe that using a combination of light and dark incubations, will allow us to define the differences in phosphorus dynamics and metabolism for distinct groups within the microbial community, so within that particulate phosphorus grouping. And I also hypothesize that by surveying like the allocation of phosphorus to that ATP molecule in ocean transects can really highlight strategic differences in metabolic processes by microbes, so different strategies of their metabolism. So how are we going to do this? Phosphorus stocks, again, are these main three pools, are main two big pools, and then break that down in dissolve to DIP and DOP, and then we have particulate P. And so for measuring the stocks or measuring the concentration of these pools, they're just done by measuring the concentration of these pools. So how we do that is we take whole seawater, and through differing phases of reaction. So for dissolved inorganic phosphorus, we just take the seawater and react it with a molybdenum blue reaction, which can then be analyzed on the spectrophotometer. For total dissolved phosphorus, we'll take all of the phosphorus and we hydrolyze it. So we take that organic pool and turn it into inorganic pool, and then we in turn read that on the spectrophotometer. So that will give us all of the dissolved pool. So through subtraction of the whole dissolved pool and the just inorganic pool, we can get that organic dissolved phosphorus pool size. And then for particulate phosphorus, we take the whole seawater, we filter it, so then we're getting the particles only, and then we do a hydrolyzation and analysis, just like we did with the total dissolved phosphorus. So this allows us to quantify the stocks of those phosphorus pools. And then quantifying the stocks of the adenylate system within the microbes, we use this fun, novel technique that we developed in our lab, Kim and I, Kim Poppendorf. So we collect the seawater. This was done from a ship. We can also collect it locally from docks, filter, and then boil those filters to then extract the adenylates, ATP, ADP, AMP from within the cells. Then we centrifuge out the filter particles. So this is now turning the particles into into dissolved ATP, ADP, AMP, and then we can analyze that using a method on the HPLC or high pressure liquid chromatography. So now I've talked about how we're going to measure the different pools of phosphorus and within the phosphorus particulate pool, the ATP, but we're also interested in the rates of change, so movement of these different pools. So phosphorus fluxes from dissolved nutrients into microbes. So this dissolved inorganic phosphate uptake is the most common, the way that all microbes take up phosphorus. Their main way of taking it up is through this inorganic phosphorus uptake into microbes. So we want to measure the size of this arrow, and that can be done by quantifying the rates of change, but that requires a time component. So what we do is we use radiolabeled phosphate, and so we add like a dye tracer to this inorganic phosphorus, and then we can trace it going into microbes. So think about if you're looking at a river, and the amount total water in the river is the pool size, and the current of that river is this flux. And we're putting a dye in the river and tracking it over time, so that way we can calculate this flux arrow of of like the river. So calculate the flux of phosphorus into the particulate pool. How we measure that is we add the radiolabeled phosphate to our seawater and do a short incubation. And during this incubation, this radiolabeled phosphate is being taken up by all microbes and incorporated into their particulate phase. And then we terminate the incubation with this tracer by filtering it. And then that now will stop the incubation. We filter it and so like there's no more free floating radioactive phosphorus or phosphorus for it to take up. We then do that at multiple time points and we get that time component and we can measure the phosphorus that was just taken up and incorporated into the whole cell as biomass. But that again does not tell us what they're doing with it. So we further analyze the incorporation of that phosphorus directly into the metabolic components of the adenylate system by doing the same initial type of experiment, incubating in seawater for a short amount of time and terminating at multiple time points on filters. But then we do that extraction process that I showed you the photos of. So the boiling in the tryst and then the centrifugation and then using the HPLC to analyze it. But now we're also going to analyze it in form of preparative HPLC. So again, if we're looking in that bottle, we have the 32 radiolabeled phosphate being incorporated into that ATP, and whether it's incorporated into the whole molecule of ATP will tell us about the metabolic rates. And then if it just gets incorporated into that AMP, that's only formed through de novo synthesis, which is growth or new production. And so that can tell us, the incorporation into AMP can tell us about the growth rates of the microbes. So on the HPLC, what happens over time, we're detecting the absorbance from this adenine molecule. And so then we can separate out, using chromatography, ATP from ADP from AMP. So the actual peak areas of these different peaks, absorbance peaks, tells us the concentration. And then we have a fraction collector at the end of our instrument. So then after it comes off at these specific times, we can collect the specific fractions for ATP, ADP, and AMP that have been radiolabeled, and then we can analyze that in order to get the rates of incorporation or the phosphorylation rates of ATP, ADP, AMP. So the power of being able to do all of the concentrations and the rates gives us the ability to do turnover rates. So a turnover rate specifically is the total inventory divided by the input or output rate of that inventory. So thinking about the, not just like the size or the amount concentration of the phosphorus, and not just like the input or output of phosphorus into the system, but the total, the turnover rate will give us the average time that something actually spends in the system. Like thinking about the river, it's not just the current that matters, and it's not just the amount of water, but it's the combination of both that tells you how long something would stay in the system. And so that's really important and powerful for understanding the metabolic processes and the pools of phosphorus that are being exchanged in the ocean. So now getting into the experiments that we did the temperature incubation that I mentioned. So this was done on the, here's Biscayne Bay, so this is at RSMAS on the docks, and we set up a control and an elevated temperature incubation. So these giant bins have seawater flowing through them, it's being pumped through them, so they're maintaining whatever the natural conditions are, but inside the bins are water that was seawater collected on the initial time point. And that way we have 3 individual bottles for each day, and then we have them again over here in our elevated temperature experiment bin. So the way that we did the elevated temperature is we had the seawater pumping through the incubator, but then we had a Nova precision cooker, so that's for cooking, yes, but it is also great for science. We can set a temperature and we can tell it to maintain that temperature, and it just keeps it at above whatever temperature the natural seawater was at. for however long you have it turned on. So we had an elevated temperature then, so all the bottles in here were subsampled on the day. So on each day, we quantified and compared those phosphorus pool concentrations, so DIP, DOP, particulate phosphorus, the uptake of the phosphorus, so that going into from the seawater into the cells, just straight uptake into the microbes of phosphorus. And then within that, the adenylate concentrations in the cells and the uptake rates, or the phosphorylation, the creation of ATP or the creation of AMP, the rates of that. And then because we were getting the pool size and the rates, we could then do turnover time. So this tells us how quickly the phosphorus was turning over in the control versus elevated temperature, or like how that affected directly the turnover in the ATP, ADP, and AMP pools, so their metabolism versus growth. So we did this over the course of 4 days. So we have the initial day 0 was on January 25th, and then each day after that, we subsampled at around 8:30, 8:40 a.m. Daniela, Mazel, and I. So we did, so these time points that we subsampled at are indicated by these dashed lines. And in those incubators, we had probes that were capturing the light and the temperature data throughout the incubation. So you can see here that our warm incubator was warmer by about 2 to 4 degrees Celsius than our ambient. And these peaks that you're seeing here are just because, again, that was like around noon each day. So that's just the natural seawater is increasing in temperature and then decreasing in temperature. And here, interestingly, we turned off the temperature probe, or the precision cooker. And then after sampling, we turned it off before we collected everything out of the incubators from the dock. And you can see the very rapid decrease in temperature to meet back up with that ambient seawater. So we were, in fact, controlling that temperature through those days and keeping it higher than ambient. So this tells us that our incubation setup worked. Now looking at what happened with the phosphorus data. So looking here, this is over time of our incubation. So from day 0, so the initial seawater conditions, and then over time, our incubators. But we're very, the question that we were interested in, again, was how does temperature impact it? So looking at that ambient, the blue line, versus the warm, the red line. So we're really looking at the differences between these two lines. And this, in all of our plots I'm going to be showing you, that's what we should be focusing on. There are other interesting dynamics that I would love to get into in the future, but not for this talk. So here we have in our first graph is the concentration of phosphate. So you can see the concentration of phosphate in the dissolved inorganic phosphate through the incubation between the ambient and warm wasn't significantly different, except in that last day where we saw a larger pool size of that dissolved inorganic phosphorus in the warm incubators. And for uptake rates, so this is the flow, that arrow going of all the phosphate going into the particles. And you can see here that, again, there wasn't a lot of significant differences, except in that last day, day 4, where we saw a much higher uptake rate. So more being pulled up into those microbes per time in that warm incubation condition. And the turnover times, that's being able to use both concentration and the rates, really tells us, like, okay, net what's happening in the system. It didn't change very much throughout the incubation overall. So that tells us something interesting was happening, where just because the concentrations may have been changing and the rates that those concentrations were taken up into, the phosphate was taken up into the cells, we didn't really see much change in the turnover, so between those conditions. So that tells us that, like, if you're turning a wheel and you have the size of the wheel is changing and the rate at which it's turning is changing. But overall, the whole wheel is net getting you there at the same amount. So it's just like the size and the rate are essentially counteracting in this situation. But we want to focus in on day 4, because that's the date where we really saw differences between that ambient and the warm condition. You can see here that on that day, we do have the DIP concentration was greater. So there's more inorganic phosphorus in that warm scenario. And we saw an increase in the uptake rate. So not only was there more inorganic phosphorus, but a larger arrow, more of that phosphorus was taken up into cells per minute, per hour, into that particulate phase. But overall, we do see a slight, very slight difference, may or may not be significant, in the actual turnover rates of the phosphorus. So the amount of time that the whole pool of phosphorus, the average time that phosphorus was remaining in the seawater, because DIP, dissolved inorganic phosphorus, in the seawater was not changing very much. Only slightly faster was the seawater actually being turned over in the inorganic phosphorus pool. So the phosphate was overall not really changing amount of time that you have in that inorganic pool. But we want to look at more specifically, how did they change intracellularly? What was metabolically changing for the microbes? So looking at the adenylate system. So this is the phosphorylation rate. So the production of ATP, ADP, and AMP over time of the incubation. Again, the ambient is the blue line and the warm is the red line. So we're focused on the differences between those two conditions. And we can see that again, not much was different until day 4. So that's really telling us that the impact of temperature, it took a while for the microbes to have anything metabolically change until due to that increased temperature. And again, ATP and ADP are more, those are the ones where you have those phosphate to phosphate bonds that have energy. So those are the ones that are really impacting your metabolic energy, versus AMP is changes in growth. So you can see here that we're not really seeing much change between those two conditions in AMP due to growth. And so due a change in their rate of creation of AMP, but ADP and AMP, we are seeing those increases. And if we zoom in on day 4, where we can really focus on the warm versus the ambient, we can see here that the ambient had a lower rate of creation of ATP than the warm. So the warm was creating a lot more ATP overall, a lot quicker of creation of ATP than the ambient, and same for ADP. But there was not a significant change in the growth between the warm and the ambient condition. This tells us that the temperature impact did not necessarily impact the growth in the same way that it did metabolism, which agrees with our hypothesis. So if we're looking specifically at those turnover times, like I mentioned, that tells you how long something actually stays in those pools. So how long is ATP staying in the, or phosphorus staying in that ATP pool versus the ADP pool versus the AMP pool? This really tells you how quickly the whole ATP system is turning over. You may notice that some of these are negative, and that's because the rate of ATP pool turnover was so fast that it was quite difficult for our methods to quantitatively analyze it, but it's just like you can see that it's very, very quick. And also it's hard to tell what's significantly different between the two pools of, or the two conditions, ambient and warm. For ADP, it's also indicative of those metabolic rates. It's just a little slower because it's a more complex production of the molecule. And so you have here that there's not a lot of differences, but there is some in the day 4 condition, where warm was lower turnover time. So that tells you it's faster at exchanging those rates of change versus the ambient. And then for AMP, there's quite a lot of change over time that may be drawing your eye, but again, we're looking at the differences between these conditions, and overall, there's not a significant difference between the warm and the ambient condition. So zooming in on that day 4, we have very, very quick ATP turnover, so very small turnover times, very quick exchange of that whole pool of ATP, and the ADP gives you a little better view that there was a significant change in the quickness of those pool turnovers with the warm condition. So we had a lot faster metabolic turnover rates with the warm condition. So that temperature really changed our rates of change, which was again our hypothesis, because you have temperature directly impacts the microbial community, especially because they are single cells, and temperature will increase the rate of enzymatic enzymes, their rate of changing different molecules in cells. And so the temperature is hypothesized to have a very direct impact on their metabolism. But all microbes, have different growths that they are different temperatures at which they have they're idealized to grow, their optimal growth temperature, and they have more control over their growth rates potentially, or that's hypothesized. And now we can directly see that, yes, we saw that change in the metabolic turnover with the temperature that we did not see in the growth. There's not a significant difference in the growth with the temperature raised versus the ambient. So what do we hypothesize was really going on overall? Because I've talked to you about the phosphorus fluxes and within that, the ATP and the adenylate changes. So here we have the dissolved inorganic pool being taken up by microbes, and we traced that, all the pools and the flux using the 32P. And what we hypothesize is that all microbes also produce extracellular enzymes, and these enzymes again are going to be increasing in their rates of change with temperature. So these extracellular enzymes will be directly impacted by the increase in temperature, and they specifically are used for hydrolyzing organic phosphorus into inorganic phosphorus. So that means that these enzymes are going to be potentially increasing the rate at which the organic phosphorus is changing into inorganic phosphorus, which would then alter the pool sizes. So now you have the organic phosphorus pool sizes decreasing the dissolved organic phosphorus, and the inorganic is increasing. So we did see in that day 4 that increase in the inorganic phosphorus pool size compared in the warm compared to the ambient temperature. And so further, that would increase, the microbes would then also increase their rate of uptake by the phosphorus. So they're increasing the amount of phosphate they take up into their cells. And so that would result in an increase in the DIP stocks, an increase in the uptake without changing the turnover time of DIP. And that's exactly what we saw with our data. So what this is saying is that it's being able to measure both the stocks and the rates is really highlighting all the dynamics that are going on. And again, this is our hypothesis of what was actually happening. We still need to finalize. We have the total dissolved phosphorus pool samples, which would then give us by calculation the organic pool size. Those samples still need to be analyzed. And then we do have the actual total phosphorus concentrations for the particulate pool that also needs to be analyzed. So once we analyze those, we can really get even more distinct processes that were going on in the elevated temperature versus not, and how that was changing the pool sizes of these phosphorus dynamics and their impacts on the metabolism, which really tells us again how that's changing all other cycles in the ocean that these microbes are partaking in. And so again, let's zoom in on that particulate pool. So we have two main types of microbes in functional groups. You have your microbes that generate ATP through photosynthesis, so your phytoplankton, and you have those that do not, so your heterotrophs. And these two have very functionally different methods, because the production of ATP through phytoplankton just uses light, and so theoretically could have very high production during light, and the other microbes are not quite so directly impacted by light. So we want to separate out the functions within these microbial groups, and to get even more diagnostic understanding of their roles in these metabolic cycles. The way we could do that is using the light versus dark incubation. So with a light incubation, we're going to capture all of the metabolic activity with all of the microbes together. And so that includes the phytoplankton. And then in the dark, we're going to essentially be eliminating photosynthesis, therefore eliminating that production by phytoplankton. And we're going to be focusing on the non-photosynthesis microbial activity. So we can then differentiate between the two groups. And we hypothesized again that we would be able to do this with the light and the dark incubations. So how we did that is we again had our seawater collected, and then we incubated for 5 hours after collection in the light for, so using a grow lamp, so they still have the full spectrum of light that they need. And then in the dark, so what we did is we wrapped bottles in electrical tape and then covered them in foil to make sure there was as little light as possible potentially getting in. And we incubated them for 5 hours, and then we added that tracer to trace the metabolism. So here we were focusing on just kind of like a proof of concept of that this would work. And so we didn't analyze all the different pool sizes, but we did analyze the inorganic phosphorus pool size and before we did the incubation, and then we analyzed the adenylate pool size, but also before the incubation. And so we're focusing mostly on the actual fluxes, the changes in fluxes for this proof of concept. And then we, in the future, will analyze with everything. So what happened? We had a change in, we did not have a change in the phosphorus rates. So here you can see the dark incubation bottles on the top in the purple, and then the light on the bottom. And this actually matches with what we would expect to happen, because this is a brief incubation, so we would not have expected much change in the actual phosphorus cycling dynamics between these two conditions, and that's what we saw. So that tells us that we were not actually changing the processes of phosphorus uptake into the cells. So now what we see in the adenylates would actually be changes in their measured metabolism between these two conditions. So we did see an increase in the ATP uptake rate, or ATP phosphorylation, so creation of ATP in the light versus the dark. So that means that we were capturing, again, that whole community. So it makes sense that the whole community has more production of ATP than when you're looking at a smaller portion of the community. So this tells us that we were indeed capturing the whole community versus not including photosynthesis in that dark incubation. And when we look at the turnover time, the amount of time that the community was churning through ATP, so how quickly that phosphorus was going into ATP and then being released on average as a whole. So this tells us that the light condition was turning over much quicker than the dark condition. And as you can see, these times are like about a minute to a minute and a half. So this is very quick for both, but that tells us that the light condition and the ability for photosynthesis was a much faster turnover of the metabolism than in the dark condition, where we have only the heterotrophic bacteria that have to take up or heterotrophic activity. So there's taking up from a different carbon molecules for energy, and you're not producing it via photosynthesis and light. For this kind of, our hypothesis was met. We can differentiate our two functional groups within the microbes using this light dark incubation. To get further into this, we're going to need to do more studies, hopefully over a Adenylate cycle, to really see how those two groups were functionally changing independent of each other over a day cycle. So moving on to my last environmental study. So we studied, these were all that we showed you so far, were all done, experimental incubations done here and at Biscayne Bay. And so I did do an environmental survey out in the South Pacific. So this is going from Tahiti to Chile, where we measured these natural concentrations, so the natural pool size of ATP across the transect and the total, all of the other pool sizes as well. So dissolved inorganic phosphorus, organic phosphorus, and the particulate phosphorus. And so we were able to analyze the functional differences of groups across this transect based off of the amount of that ATP per cell that we saw. So what that looked like across our transect is, again, we have the total particulate ATPs, the ATP in cells, and out of the total, this is normalized to the total particulate phosphorus that we saw or measured. So this is the allocation of that phosphorus into the ATP pool rather than into other biomolecules such as lipids, DNA, RNA. That kind of tells you the prioritization of microbes to having these high energy molecules which are used for the for metabolism. But we were not able to measure rates due to limitations with using radioisotope tracers on cruises, and we didn't even have them at the time. So we were interested in this pattern, like we can see here that in these deep microbes, and especially on this eastern portion of the transect, so closest to Chile, we have an increase in the amount of ATP per phosphorus molecule in these cells. So we looked at all the other data that we had, and we're trying to figure out what could this possibly be tied to. So initially, looking at depth, you can see that this line here where we have microbes deeper than the, this is below the euphotic zone, so subeuphotic microbes. It also coincides with having a decrease in dissolved organic carbon. So if you remember, I said microbes all need three main nutrients: carbon, nitrogen, and phosphorus. So in this area, you're going to have much less frequency of microbes being able to access the carbon that they need. And so this, we believe, could be changing the microbial strategies of them or of their metabolism, so that they're creating essentially a battery pack of ATP. They're storing up on ATP, so when they do interact with carbon, when it's available, they're able to then use it to perform other metabolic necessary functions, like for DNA, RNA growth, other production rates. So we think that having that low DOC may be changing their strategies in the subeuphotic area, and therefore increasing their ATP stocks. Alternatively, we were also, some of you may know that in this going across towards Chile, there is an oxygen minimum zone. And so here you can see the zone going deeper than what we were measuring. So again, this is from west to east, so going across this transect with depth, and you have this increase here in that ATP per phosphorus cell that also coincided with that change in oxygen concentration, that stark decrease in oxygen. And microbes in that zone are now going to be using anaerobic metabolisms, or the microbes there use anaerobic metabolisms, which make them have a very different metabolic system, and therefore may result in changes in that pool size of ATP, which were not present in the aerobic metabolisms of the microbes. So this really highlights that ATP could be indicative of these strategic changes by the pool size changes in the microbes, other than just the rate changes. So in conclusion, our method, so the ability to measure these adenolates, is allowed for direct measurement of microbial metabolic responses across diverse environments and different conditions. So we observed in our temperature incubation a direct increase in the metabolic turnover rates of these microbes within their phosphorus uptake after 4 days under elevated temperature. So we saw that they were changing that metabolic processes independent of their growth rates. We did not see a significant change in the growth rate between the temperature increased and the ambient. So that our hypothesis for that was confirmed. In the combined light and dark incubations, those incubations enabled us to differentiate the two main metabolisms of the light, so photosynthesizers and non-photosynthesizers. Again, we will be doing more, hopefully, incubations to get more into those differences. And we also completed an oceanographic survey that distinguished different metabolic strategies in microbes in the decreased oxygen and with depth based off of the ATP as a fraction of the total particulate phosphorus. And so that really highlights that even just looking at the pools when rates aren't enabled, being able to analyze the adenylate concentrations can tell you a lot about their potential strategies. So in the future, we would like to implement this adenylate system measurement, both the stocks and the rates, and couple it with other measures of microbial community dynamics to better understand their roles and impacts in those biogeochemical cycles and the microbial loop. So better understanding how they're impacting carbon sequestration into the deep ocean and export and production and consumption of oxygen and nutrients. And these other measures of the microbial community include transcriptomics and microbial abundances and their community composition to a taxonomic level. That's really what the power of this is. We can incorporate it with any of these measurements for any microbial community, because again, the adenylate system is ubiquitous. And using this as a study or a way to study marine microbes will also provide a new way of looking at the flow of biomass and energy in the ocean, which is a lot of biogeochemical cycling and what we're really key and interested in understanding better. So with that, I would like to acknowledge so many people have been a part of all of this work. Lately, Daniela has done a lot of work helping me collect all of those seawater samples for all of those incubations that we've been doing locally, and other people in the lab, in the Poppendorf lab, have really helped. And then I've also, my committee members, Kim Poppendorf, Dennis Hansel, Larry Brandt, and Hillary Close, and all of the crew and everyone from the RV, Nathaniel B. Palmer, here's a picture of it, from that transect that we did in the South Pacific, as well as all of my funding sources. And with that, I would like to take if anyone has any questions.
All right, great presentation, Kaycie. Thank you for giving it. Everybody give a round of applause.
Thank you.
All right, so if you have any questions, you can unmute yourself and turn on your video. Otherwise, you can ask them in the chat and I'll read them.
Hey, Kaycie, this is Dennis. I have questions. How you doing? Great job. Great progress over the years. It's amazing to see what you've been able to accomplish. Congratulations on that.
Thank you.
So, when you were talking about those, the light dark experiments, I was wondering what would you see in the mesopelagic ocean if you could eliminate, those photoautotrophs, and would you start to resolve growth versus metabolism in those organisms that are surviving on the export flux of particles and solubilization of DOC? Would that, it seems to me that'd be useful if you could do that, because right now you just have, thymidine uptake, leucine uptake, things like that, and very narrow view of the system.
Yes, exactly. That is something that is very key and very, like that is definitely something we can use this method for, and incorporating that with the dark and light incubations and doing that with these other measurements in the across transect. So I think that would be definitely something that can be done. And I think in doing so, we would be able to look at specifically the metabolisms that were not due to light. So having the dark incubations alone would enable us to look at the turnover rates and metabolic versus the growth rates of those microbes. So yes, that is definitely something we could do.
Yeah, just it's, you know, I'm not a microbial person, but it just seems to me that in the mesopelagic, you know, it seems like the organisms there are taking advantage of substrate that's available to them for sustaining themselves, but not necessarily for growing, right? Their growth seem to be extremely low down there. And, you know, if you could resolve that or show that with your incubations, that'd be a nice thing.
I think that's actually like, yeah, that exact, so I know that Kim has been looking into or submitted proposals to try and actually do that on a cruise in California Current, and being able to also incorporate direct growth measurements using the phosphate uptake, that radiolabeled uptake, and incorporation into lipids directly can tell you the growth rates of heterotrophic bacteria specifically. So then we can use that incorporation of phosphorus into lipids for bacteria growth rates, in addition to the phosphorus incorporation into the adenylate pool to get that metabolic rates. And so that would even more specifically like show us exactly how those growth versus metabolism would change, and doing that with light and dark incubations would further highlight the differences between autotrophic bacteria versus heterotrophic bacteria.
Great. I've got one more question, and I've got to leave by noon. I got another Zoom call. So in your section plot, you know, particulate ATP in ratio to the particulate phosphorus, right? And you had your max right there in those low oxygen waters, you know, along the margin there. Yeah, right there. Yeah. So those waters with the elevated ratio would also be the ones that are feeling the most export, you know, you know, that's the most productive waters. And so you're in the upper mesopelagic there. And so did you, you know, I think you gave two scenarios that could explain it, but could it just be a response to the very high export production and them taking advantage? You know, they're not in euphotoxone, but they're getting a lot of organic matter right in those waters.
Yes, I do. We did. So if you can, I know that the, I'm trying to think through my, so we did consider that. I believe that there was just the differences between the high organic matter in these regions. There were also other regions of this transect that had high organic matter but did not have that, because we're not looking at fluxes here, it's just the stocks. Right,
but they're experiencing fluxes there.
Yes, exactly. We would love to have measured fluxes there.
Yeah, I just mean the sinking flux there is going to be very high relative to anywhere else, you know, on that line going west.
Yes.
Those waters, you know, they're low oxygen, but independent of being low oxygen, they are also receiving a lot of downward flux of organic matter
from the surface. You mean
from the euphotoxone just dropping down? And so, you know, if you had some measure of you know, export production across that section there, you'd see real high rates over there in the east and low rates as you go west. And so, you know, it could be just a function of that export production right in that area.
Yeah,
just something I'm
definitely
sure you thought about it, but I didn't hear it. So
yeah, unfortunately we couldn't measure that with this, but yeah, that is interesting to think about. Is there just interaction with that export versus the, I mean, we do have the dissolved pool concentrations here, so there is not a ton of carbon that was measured in that export.
But you know, the sinking part, you know, release DOC and it's used, so it doesn't accumulate much. a little bit, but only for a short time, and then it's consumed. So anyway, good job.
Thank you.
All
Oh,
right, do we have any other questions?
I see Cameron.
Oh, there's a little baby.
Hi, Cameron. Oh, I like your little hat, Cameron.
He always gets very excited when we talk about AMP.
All right, everybody, if that's it, let me scroll through here. Yep, okay. Thank you for coming today. We'll see you next Wednesday for Compass Seminar again.
Great job,
Bye,
Kaycie.
everybody. Great job, Kaycie.
Thank you.
Really nice job, Kaycie.